Hey, what is going on? It is Thursday, March 3rd, 2022, and I am excited to be here today to talk to you about, or rather, continue on talking about the cell organelles, because my goal is to get down to the DNA itself and really delve into the genetic code, but we must wade through the soup of organic material which lives in the cell, which all kind of is involved in cellular function and which supports the whole process of DNA synthesis and DNA replication and all the other parts of the so-called central dogma of biology, right? Because a lot of times in school you kind of skip down just to the DNA and you're like, oh, okay, this is important, but it's really they kind of gloss over until you get to the higher level biology and cell biology classes and organic chemistry. They gloss over a lot of those subjects in like high school and whatnot, but we really need to eventually delve deep, and that's my little precursor. So I know you want to hear about the peroxisomes, right? That's what I have on the screen. I was going to talk more about the lysosomes. I did leave off on my last video on a few of the lysosomal functions in autophagy, and that's something that I'm going to delve into significantly, but I'd rather get to autophagy when I talk more about apoptosis and other other processes that autophagy is involved in, right? So anyway, so today I'm going to skip to peroxisomes because they're really important. They're very, very important organelles. And actually, I think in school, in high school at least, or even earlier, they kind of they again gloss over this organelle. It's not. I think it was actually its function was discovered more late in the whole stack of different organelles that had been described in classical biological studies. But either way, it serves a very important function. So it's basically this spherical organelle. A lot of the organelles are spherical. But this is a spherical organelle. Again, it has an endomembrane as a plasma membrane, like most organelles do, and a lot of what happens in the peroxisome, there's a lot of things that go on inside of it, and it's mostly responsible for. Let's see if we can list them: free radical, free radical annihilation, which is super important because many of your biological processes, including the electron transport chain in mitochondria, as a result of or involved in the Krebs cycle, results in the creation of free radicals. And remember, for those of you who are not familiar or don't remember too much about what free radicals do, free radicals are ions essentially, which are unstable and they want to bind to your essential macromolecules. Like they want to bind to your DNA, right? They want to bind to your fatty acids. They want to bind to proteins, and if they bind to any of these things, right? If they bind to your DNA, for instance, that even you need an elementary knowledge of biochemistry to know that's bad and that will cause mutations in your DNA, damage to your DNA, which could cause mutations during translation, which we'll get to. Free radical interaction with proteins is bad because they change the protein function, right? The protein's form is required to function in a specific way. The form informs the function. So if the form is damaged, the function suffers, right? And so you get diseases as a result. And essentially, free radicals, also at one point in time, free radicals was one of those hot buzzwords, and it still probably is when it comes to anti-aging and whatnot, because it is considered to be one of the components of aging because it leads to cellular damage, and free radical accumulation can occur later in life through many reasons, but it definitely occurs in your body through the result of your standard life processes anyway. So, example of free radicals that I just want to talk about is hydrogen peroxide, right? Hydroxyl molecules, superoxide anion, right? These all, like, if you look at how they interact, these will uh, cause, and these are just sources of free radicals, but like, like there's like, we can go find a list of them somewhere, but they will cause oxidative stress on your cells, and they will damage everything, all your organelles, etc. So I took a detour, but let's go back to peroxisomes. So peroxisomes break down free radicals. They have processes in which they do that specifically. They also deal with alcohols and ethanols, right? So ethanols, alcohol has to be broken down somehow in your cells, and alcohols are not only things that you ingest, right? They're also they're also byproducts of several processes within your cells, metabolic functions. So, it's not all just drinking alcohol, right? So peroxisomes also are involved in that process. Peroxisomes are also involved in beta oxidation of very long chain fatty acids. Now that is a mouthful, right? And in order to talk about beta oxidation is also a very core life process, and it occurs in all cells, and it occurs in prokaryotes in their cytosol, and it occurs in eukaryotes in the mitochondria, in the peroxisomes, in the smooth and the plasmic reticulum, which we talked about. But the beta oxidation, we can jump to that. Beta oxidation, just to remind anybody who has their organic Chem 2 knowledge down, is a process by which fatty acids are broken down and they are turned into acetyl-CoA, right? I'm going to go slowly here. They're turned to acetyl-CoA, and then acetyl-CoA, this is actually a better diagram of the full, forget the tumor cells for a second. The acetyl-CoA then is transported to the mitochondria through several intermediate accessory proteins, transporter proteins, etc. And then the acetyl-CoA powers the Krebs cycle, which then acetyl-CoA is not the only thing that's involved here. There's other things, but it powers the Krebs cycle, which then leads to ATP production, right? Krebs cycle, citric acid cycle, TCA, whatever you want to, the citric acid cycle, TCA, whatever you want to call it, right? That cycle is the only thing that allows us to move, right? Like, we wouldn't have enough ATP through glycolysis, which is something all living things pretty much have in order to do our daily function. So us as advanced organisms built on eukaryotic cells need the Krebs cycle to produce that ATP, and that's learned in fundamental biology without a doubt. Though all the intermediate products are probably not, unless you go to like a school in Japan where they teach all that stuff. But anyway, we're not going to go into the Krebs cycle right now, and that will be something we go into later, but that's kind of old hat. I'm just jumping back to beta oxidation because one thing we have to know is that, or one thing we should wonder about is, why does beta oxidation take place in the peroxisomes, the smooth ER, and the mitochondria? Okay, because it's so important. That's one thing. It happens everywhere. But the peroxisomes do it and why can't, another question is, if acetyl-CoA eventually goes to the mitochondria, right? Why can't the mitochondria just handle all of beta oxidation? Well, what happens is sometimes your fatty acids are very long, right? So like if you remember, very long chain fatty acids. Acids are one of those macromolecules which are kind of like fatty acids, which are kind of like proteins, which kind of have like they have a polymeric structure, right? So they can they are not defined by a specific molecular formula. They have repeating subunits, right? And they can get arbitrarily long. So when you ingest these very long chain fatty acids like this here, depending on how long it is, it may be too big to enter the mitochondria, and that's important, right? So in order to enter the mitochondria, we have to first break it down into acetyl-CoA and into acyl-CoA subunits, right? So now, one thing to remember: there's acetyl-CoA and then there's acyl-CoA, right? And acyl-CoA, essentially, CoA is the main part. Coenzyme A is involved through another process which adds coenzyme A to fatty acids, which allow them to be broken down through repetitive oxidation cycles into acetyl CoA, and then those finally, the acetyl CoA, after it's broken down sufficiently, then can enter the mitochondria through several mechanisms, right? Getting too deep into that is going to be pretty crazy. We have to talk about carinite transporter, protein packages and such, but let's not go down that rabbit hole for now. Let's just know that that's what peroxisomes do, and peroxisomes essentially, now we have to talk about, let's just jump to the other mechanisms of other functions of peroxisomes. Peroxisomes also have a very important, we talked about the alcohol degradation, not only fatty acid degradation, we talked about alcohol degradation. That occurs through an enzyme called catalase, okay? So catalase takes, or rather hydrogen peroxide. Catalase takes hydrogen peroxide, breaks it down into H2O plus O2, and also evocates hydroxyl as one of the products, right? Which then have to be essentially through the use of several intermediate mechanisms. This is a free radical, hydroxyl ions, right? So you need antioxidant something to convert that and to get rid of that, turn it into water, etc. So, but basically catalase essentially reduces what's called xenobiotic toxicity, right? And this is like essentially, like, this is a small diagram, but essentially it leads to the degradation of oxygen, or sorry, so the degradation of alcohol, right, and the neutralization of the effects of alcohol, right? So if you don't have so there are diseases like autosomal recessive diseases, like something called Zellweger syndrome, a few other diseases which lead to and which essentially they're recessive, so they need two mutant alleles, one from each parent, or they need to be combined in that way, such that the genes, specifically a gene called PEX gene, is knocked out. And once PEX gene is knocked out, then essentially your peroxisome production will reduce. And when your peroxisome production reduces, you get loss of function for a metabolism, for beta oxidation reduces, so you accumulate very long chain fatty acids. You also accumulate branch chain fatty acids, which is another thing to talk about, branch fatty acids at some point, but they're essentially, indicated by beta oxidation. You also lose this ability to get rid of free radicals. You lose alcohol. You accumulate free radicals. You accumulate ethanol if you drink it, and essentially this accumulation of this material, which normally gets broken down and converted to energy, then starts to affect and damage your organs, because your body is supposed to get rid of these and supposed to produce this energy. So you can imagine all the effects of this, throughout your nervous system, your renal system, many systems in your body will suffer as a result of Zellweger syndrome or peroxisome deactivation, or lessened peroxisome activity. So that's just a little bit on the peroxisomes. We're going to eventually go deeper into them at some point, but it's another step between the next step to getting deeper within the cellular genetic function. Okay, so I'll talk to you later. Have a nice day. See you around.